See you.
Oh, now,
Oh, there you
Oh,
are,
hey,
George.
Shilva.
okay. George.
Chacha, your beautiful face we can finally look at.
Okay, with that, Arun,
All
Arun
right.
doesn't need
Thank
any
you.
introduction, so
Yeah.
I will let Arun take over the mic and
Okay.
start his presentation.
All right.
Thanks,
Thank
Arun,
you.
once again for
Yeah.
giving your talk to
No,
this
no, no. Thank you.
feedback.
Thank you, CS. It's my pleasure. And so today I have a short story to share with you. But before I do that, I just want to thank again CS. He has been always very nice to me, very kind to me. And I was really touched by his email that he sent out, uh, saying nice things about me. But so I just want you to lower your expectations. So I don't want to disappoint you. So he did make one slight, uh, uh, I don't want to call it error, but I'll correct that, whatever you may call it. So let me just start with the. So he said this would be my third presentation. Can you see my slide?
Not yet.
Oh, okay.
Okay, yeah, now it's coming.
Now you can see. Okay. So actually, this is going to be my fourth one. Can you see my full screen or no?
Yeah, you can see
Yeah.
your
Okay.
screen. Yes.
So actually, the very first presentation to this distinguished colleagues of mine I gave was I introduced AAV, which I call AAV has gone from almost a virus to an awesome vector. And I gave this presentation on May 22nd, 2021. So that was my first one. And the second one was where I talked about the use of AAV vectors in phase 1-2 clinical trials in the US and hopefully soon in India. This trial will start hopefully in January. And this presentation I gave on September 25th, 2021. And then last year, CS gave me an opportunity to talk about my journey from starting with Avigen, which I'll talk about in a moment, to Navigen, the second company. And this took a very long time. So I titled it The Long and Winding Road. And this was on October 21st of last year. So today, which is November 16, 2024, and I mentioned earlier, those of you may have missed it, I definitely cleared it with CS to make sure I don't offend anyone. So the title of today's presentation is Yes, We Can. All right, so what is it about? So some of you may not have heard any of the previous talks. I'm just going to very briefly describe what AAV is, and so some of the slides may be, but just a few slides you may have seen before, so please bear with me. So before I do that, this is a very auspicious day in my life, August 16, 1973. For those of you who think that's my birthday, thank you very much. No, that's not my birthday. That was the very first day I actually came to Bangalore and joined as a grad student in the Department of Microbiology. It used to be called MPL, Microbiology and Pharmacology Laboratory, and then it became MCBL. So this was my first day. It was the most exciting day for me. I was thrilled to be there. And then spent 1 year, and the last week of my first year, this paper was published. So on August 9, 1974, a little over 50 years now, by a guy named Thomas Cavalier Smith. He was at King's College in London, and he published this he was a theoretical biologist. So he published this paper, Palindromic Base Sequences and Replication of Eukaryotic Chromosomes. Can you see my, let me see if I can.
Yeah, it's clear.
You can see this? Okay. So, he actually hypothesized that if there is a single-stranded DNA, how would it replicate? And he came up with a fantastic mechanism. This is all theoretical. So he said that no DNA polymerase can initiate DNA synthesis. It can only extend a pre-existing 3' end. So he came up with I was blown away by this paper when I read this. So he said the 3' end has to have a palindrome. So in other words, it will actually fold over itself and create a primer. And now the DNA polymerase can simply extend this. The problem is then how do you resolve this hairpin? So he came up with another terrific idea that you make a cut exactly opposite where the DNA synthesis began, and it will create a new 3' end, and then you simply copy it. So this completely changed my life when I read this paper. So I actually went to Dr. Gopinath. I was so stupid, I still am, but I asked, is the human DNA single stranded? So he said, of course not, it's all double stranded, right? So I actually looked up Dr. Cavalier Smith's biography, and there's a Wikipedia page on him. So if you go and read this, this is what it says. Oh, by the way, that's Dr. Cavalier Smith. So his Wikipedia says that Professor Cavalier Smith, he was then at Oxford University, has produced a large body of work which is well regarded. Still, he is controversial in a way that is a bit difficult to describe. The issue may be one of writing style. Cavalier Smith has a tendency to make pronouncements where others would use declarative sentences, to use declarative sentences where others would express an opinion, and to express opinions where angels would fear to tread. In addition, he can sound arrogant, reactionary, and even perverse. On the other hand, he has a long history of being right when everyone else was wrong. To our way of thinking, all of this is overshadowed by one incomparable virtue, the fact that he will grapple with the details. This makes for very long, very complex papers and causes all manner of dark murmuring, tearing of hair, gnashing of teeth among those tasked with trying to explain his views. So it aptly describes Dr. Cavalier Smith, really a brilliant scientist. So why am I telling you about him? So when in that paper, he talked about single-stranded DNA, right? So I was curious, is there any organism? Remember, I was a first-year grad student. I had no idea. So is there any organism that contains single-stranded DNA as its genome? And sadly, Dr. Cavalier Smith passed away 3 years ago on March 19th. Okay, so just to bring you on the same page, again, I think most of us has experienced common cold or flu-like symptoms and fever, right? Conjunctivitis, which is very common, pink eye it is called, or bronchitis, chest cold, or sore throat, right? All of us have experienced. So these are the symptoms that are caused by a virus called adenovirus. The name adenovirus is derived from the fact that the virus was actually isolated for human adenoid tissues with our, tissues that are back of our tonsils. And actually, this paper was published in 1953, December 1st, by Wally Rowe and his colleagues at the NIH. They took a picture of the adenovirus preparation, and this is the EM picture of the adenovirus. So this is a about 100 nanometer diameter particle, a large virus. And this was the first time adenovirus, had been visualized under the microscope, electron microscope. Now, one thing you notice, that's this little guy here, and this is not an arrowhead. This is just the artifact of the EM picture. And of course, this was completely ignored. Okay, so they didn't talk about this. Now, the good thing about adenovirus is that it's an extremely well-studied virus. A lot of molecular biology knowledge has come from studying this virus, and this virus can be grown to very, very high titers in the lab. So why am I telling you all this? So then these three guys, Robert Atchison, Bruce sorry, let me get this. Bruce Casto and William Hammond, they were working with adenoviruses. And as I said, these can be grown to very high titers, but they noticed that their titers took a nosedive, and they could not explain what was causing the loss of high titers of adenoviruses. So they decided to take an EM picture of their preparation, and again you can see this is the large 100 nanometer particles, which is adenovirus, but they also saw a whole bunch of smaller particles. So they came up with two hypotheses. One hypothesis was that this is something is breaking down the adenovirus particles. So these are broken down subunits of adenovirus. Or the other hypothesis was, no, these are actually building blocks of adenoviruses, and something is preventing these subunits in coming together to form the full size adenovirus particle. It turned out both of these hypotheses were completely wrong. So this is a completely new virus that seems to like growing in the presence of adenovirus. And in the process, it simply takes over and begins to inhibit adenovirus growth. So this virus was, so that was the hypothesis. These are either broken down peptide particles or they are the subunits that fail to assemble. So anyway, this virus was then called adenovirus associated virus because it was discovered as a contaminant of adenovirus. And later on, it was shortened to adeno associated virus. So this is a virus that cannot replicate in the absence of adenovirus. But once it gets going, it will begin to inhibit. So people have described this as a virus that, it's the virus that bites the hand that feeds it. So because it's very ungrateful virus in that sense. So this paper was published in 1965 in Science. So AAV was discovered in 1965. Okay. So as I said, I was curious, if there's a virus is there any organism that contains a single stranded DNA? And of course, Dr. Cavalier Smith did not mention anything about AAV, because remember, he published that paper in 1974, and AAV was discovered in 1965, so almost 10 years earlier. So this was surprising that Dr. Cavalier Smith did not mention that AAV, because he obviously didn't know. So this is Dr. Ken Burns, and one of the great claim to Dr. Burns is that he figured out that AAV actually contains a single-stranded DNA. So this was like dream come true. I said, wow, I want to work on AAV. This was in 1974. So I wrote to Dr. Burns. I said, I want to come to your lab because I want to, test the hypothesis that AAV actually can self-prime and replicate the way that Dr. Cavalier Smith hypothesized. So he said, now finish your PhD and come as a postdoc. So I said, okay. So that's what I did actually. Okay. So so this is a AAV. It says very small, 25 or less, 25 nanometers diameter. Naked icosahedral particle contains a single-stranded DNA. The only difference is that instead of the simple hairpin, it contains a T-shaped hairpin at both ends. So these are called inverted terminal repeats. So instead of a simple hairpin primer, now we have a T-shaped hairpin primer, and now the virus will replicate exactly as what Dr. Cavalier Smith had proposed. So by the time I got to Dr. Burns's lab, he had already figured out. So Dr. Cavalier Smith was absolutely right. AV replicates exactly how he predicted. So, so as I mentioned, I ended up as a research associate in his lab in 1980. But, it kind of sounds dumb to say it now, but I thought this was a dead end. AAV nobody took it seriously. I was only fascinated by the replication model, but since he had already solved that problem, I said there was not much left to do. So I only stayed in his lab for 2 years. And So I'm just going to fast forward now to a few years. And this is my disclosure slide. So yes, I did work on AAV for only 2 years, but fortunately I decided to stick with it. And today, this morning, I actually tried to figure out. So it's been 44 years, 4 months, and 15 days that I have worked on AAV. Okay, so what have I done? But I've been very fortunate that my research on AAV has been continuously funded for 42 years now. It will be 42 years when my last grant runs out in 2029. So I've been extremely fortunate. So what is AAV good for? And very early on, there was so I have a little bit of industry experience as well. Back in 1992, I was also very fortunate that I became the founding scientist of a very first AAV gene therapy company called Avigen. But my association with Avigen was only very short-lived because the CEO, and I joke about it now, we had a CEO who could not spell AAV. He was only in it for making money, and he actually took Avigen public and made a whole bunch of money. He was at that time was the most. I guess he was the wealthiest CEO among biotech companies. And I said, no, this is not for me. I talked about this in my last presentation from Avigen to Navigen, and I won't bore you with all those details. So I sat out for 25 years and stayed focused on the basic molecular biology of AAV. And 25 years later, a second company was formed, and I talked about this previously, called Lacerta Therapeutics. Again, I don't have much luck with the CEO. CEO actually drove this company into the ground, and this company was dissolved last year. So, and I was not really involved, except that I was a co-founder. So then I decided to do it on my own. So last year I started a company called Savian Therapeutics. Because when Avigen was started, we didn't even know how to make AAV, let alone how AAV would be seen by the human body. So I didn't think we were ready to do clinical trials. And the CEO wanted to take this into the clinic, which I completely disagreed. So that's why he threw me out of Avigen. So anyway, last year I started this company called Savian Therapeutics, and I talked about this previously. So I won't say anything beyond that. And last presentation I talked about launching a company. I recently launched this company called Navigen Therapeutics. This is for nucleus-free genome editing. And again, I won't mention this except to point out that I think we are having a lot of difficulty with nucleases, CRISPR-Cas9 and base and prime editors, but we think we have an alternative approach that might be safer. Okay, so the problem going back to AAV in early days, from 1965 to 1984, nobody was interested in AAV because it doesn't do anything. It doesn't replicate, doesn't cause any disease. So there was no money in AAV. So it was considered the most boring virus. All that changed in 1984. It was also dubbed almost a virus. All that changed in 1984 when Dr. Nicholas Bijiska's lab here at the University of Florida actually created the first AAV vector. So basically what he did was he replaced the AAV sequence, the coding sequence, and put in the neomycin resistance gene and driven by a promoter in the poly A. So this vector was able to confer neomycin resistant into human cells, in mammalian cells in tissue culture. So this was the first recombinant AAV vector that Nick Bizichka made. And that vector was actually created here at the University of Florida. So the first recombinant AAV vector was made here at the University of Florida in 1984. And again, fast forward to 2008, So far, AAV vectors have now shown clinical efficacy in gene therapy of 14 human diseases that has been published so far. So the first human disease that was targeted for gene therapy, I know Anand will somewhat disagree with me, but I'll say it anyway. So Leber's congenital amaurosis, which is a form of blindness, and they use AAV serotype 2, and this was published in 2008. And the first human disease was also cured with AAV vectors at the University of Florida. And again, I'm not going to read all the names of the diseases and which serotypes, and you can see this in this slide, where this is all published data. So these are the 14 human diseases that have been cured by AAV vectors from starting with 2008 up until 2024. And just this week, this paper was published in Nature Medicine, where AAV vectors, AAV-RS74 vector was shown to cure Duchenne muscular dystrophy. And this is the disease. I mean, I'm not a muscle guy, but I'm just going to focus the rest of the talk on Duchenne muscular dystrophy and AAV-RS74 vectors for today's discussion. And now there are 7 FDA-approved AAV drugs. And I'm just going to very briefly mention these. So the Leber congenital amaurosis type 2 AAV2 vectors actually became the very first FDA-approved drug called Luxturna. This is for LCA2, and this was approved in 2017, and this is the cost. So it's very expensive drug. Second drug is called Zolgensma, which is AAV9. This is for spinal muscular atrophy, approved in 2019, and this is $2.125 million per injection per patient. The next one is Hemgenix, AAV5, hemophilia B, approved 2 years ago, $3.5 million per patient. Elevidys, and this is the one I'm going to talk about today. This is AAV-RS74 for Duchenne muscular dystrophy, approved last year at $3.2 million per dose. Last year, Roctavian was also approved, AAV5 for hemophilia A, $2.9 million. Bequest was approved in April of this year for hemophilia B, again AAV-RS74, $3.5 million. And 3 days ago, there's a company called PTC Therapeutics. They got FDA approval for AAV2 vector for aromatic amino acid decarboxylase deficiency. And this was already approved in the European Union at 3 million pounds. The US price is they have priced it at $4.3 million per patient. So this is direct injection into the brain. So this is called Kebilidi. This is the most expensive drug on earth so far. So, okay. So this is fantastic, right? It has gone, AAV has now from 2008 and beyond, it has gone from an almost a virus to an awesome vector. And the prediction is that AAV vectors will cure a number of human diseases in the future. And more than 300 phase 1, 2, 3 clinical trials have been or are currently being performed with AAV vectors, and there's little doubt that we will see. But there's a saying in it probably true in every language. They say when something, and now I think Anand will agree with me, that if something is too good to be true, it probably is, right? So I hate to even say this, exactly 23 years ago, I said, I think we are doing something wrong here. It's fantastic, but this cannot be the whole story. There's something that we are missing. So, and very sad to say that, so I actually in 2001, I said, no, we need to do something to improve the first generation of AAV vectors. So I hypothesized, and this is all published, that there is room for improvement in the first generation of vectors. So why did I say that? Of course, I had no clue what's going to happen in the future. So here's a table. All I need you to do is focus on the last column, okay? So if you simply count this, now there are 10 patients who have died in AAV clinical trials. And this is very sad, right? No one needs to die. And actually one more patient died recently in a Pfizer trial. And I'll emphasize this point over and over again. So why is this happening? Why are these patients dying? So again, I'm going to pose a question to you, and I hope you will agree with me when I say that AAV evolved as a virus and not as a vector, right? No dissenting opinion on this, right? So far, no one has disagreed with me. Every time I give a talk, I mention this. So I'm hoping that that streak continues. So other thing I should mention that all of these serotype vectors that are being used, these are all first generation vectors, right? So the human body does not know how to deal with the first generation AV vectors. So again, as I said, AV is a very inefficient virus. So the first generation of AV vectors cannot be expected to be optimal. That was my main point back in 2001, that we need to make the vector more efficient. And the other point that and I'll discuss this at the very end, if you just simply look at the doses that are being given, anywhere between 1 time to 3 times 10 to the 14 vector genomes per kilogram. And that dose is extremely high. So the use of extremely high doses is what is killing these patients. This human body cannot tolerate that much antigen. Basically, AAV is a foreign virus, so body immune system will react to that. Okay, so and the other problem is, we use these vectors and we start out injecting mice, then we go to larger and larger animals, all the way to non-human primates, and then just assume that these vectors will also work in humans. And more often than not, that is not the case. Humans are not large mice or even large monkeys. So that's the point I made back in 2001, that we should pay attention to human tropism, and that was completely ignored. So anyway, after the first 10 patients died, I really felt compelled to write a review article, which was published last year. And I said, no, you need to come up with some way to overcome the limitations of the first generation of AV vectors. And I'll touch upon this very briefly. So again, as I said, I'm going to focus on Duchenne muscular dystrophy for discussion today. Okay.
Arun, just a simple
Yeah,
question.
sure.
What did they die from? Was it because the
Yeah.
disease came back, or whether
No,
it was the treatment that killed the patients?
you're 30 seconds ahead of us, so
Okay,
I'll answer
I'll
that
wait.
question.
I'll wait.
Okay. Okay. So let's talk about Duchenne muscular dystrophy using AV vectors. So 4 clinical trials have been done for gene therapy for DMD. So there's a company in Boston called Solid Biosciences. They used AV9, and they used 3 times 10 to the 14 vector genomes per kilogram. And they noticed that you can inject these into monkeys, nothing happens. So they say, "Oh, this is safe." But they noticed that various serious adverse events in these DMD patients: complement activation, thrombocytopenia, renal damage, and cardiopulmonary insufficiency. So other thing I should also mention, so we all do this, right? We inject into monkeys and then say, oh yeah, it is safe, and then go into patients. The problem is, and I wrote this, and I'll come at the very end, that we are injecting these high doses into normal monkeys, healthy monkeys. But then we go into patients who are sick. So this is not the same, right? You need to take that into account, that you cannot compare a normal healthy monkey with a sick patient. Okay, so that's trial number 1. So this trial was actually started by the FDA, put on hold by the FDA, but they have now switched the serotype and they're doing it again. Pfizer, I mentioned earlier, did the second trial also with AV9 and a little bit lower dose. 2 times 10 to the 14 vector genomes per kilogram. And this was amazing. They saw many, many more serious events: vomiting, nausea, decreased appetite, pyrexia, acute kidney injury, atypical hemolytic uremic syndrome, like complement activation and thrombocytopenia. And this is where 2 patients actually died in the trial. And this phase 3 trial also failed to reach the clinical endpoint. So Pfizer just recently fired 200 people and completely stopped this whole program. So they are not pursuing this. This was an interesting experiment that was done. There's a cure rare disease. It's actually not really a company. Well, it is a company, sort of company slash foundation. It started by a guy whose brother actually had DMD. So he was, committed to helping his brother. So they did this N of 1 trial. So there was just one patient. Again, using same AV9, one time to they lowered the dose to 1 time to the 14 vector genomes. And this was an older patient. So this is just this is to answer CK's question. So mild cardiac dysfunction was first noticed and pericardial effusion developed, followed by acute respiratory distress syndrome and cardiac arrest 6 days after gene therapy. And unfortunately, this patient died 2 days later from multi-organ failure.
And that
So
was
it's
not
not
his
the,
brother, right?
sorry, say that again?
That was not his brother.
No, it was his brother. Yeah.
It was
So
okay.
his brother was healthy brother was trying to help his sick brother. And he was an older patient. He was 27 years old. So to answer CK's question, this is not because the disease is coming back. It's just way too much vector is being delivered, and the human body cannot handle this. And I'll come back to that at the very end, and we'll see. And the 4th trial was done by another company, also in Cambridge, called Sarepta Therapeutics, and they used AAVRS74. This is a virus vector that they created from a rhesus monkey. By the way, I should mention all of these are derived from monkeys, all these serotypes, AV9 and AVR74. They went up to 2 times 10 to the 14 vector genomes per kilogram. And the only, I don't know if you call it serious adverse event, 50% of the patients had vomiting and nothing else. So this was very encouraging. Finally we have a vector that is not causing serious adverse events.
Arun?
Yes, Chandru, yes.
That's what I was wondering. You mentioned cure rare disease with only 1 patient. That former
Yes.
two also only 1 and 2 patients. The first patient died.
No, this was a phase 3 trial. There were at least more. I think, I don't know, CK can correct me, but phase 1 is a safety trial. Phase 2 is larger number of patients. Phase 3 is a much larger group of patients. So a lot of patients were dosed with this high dose, but two patients died.
This
So
may
a
be
lot
the
of
only
them.
case.
Sorry, go ahead.
So a lot of them had a better. A lot of them had a improved.
No, that's the problem. And actually, I forgot to mention, so this phase 3 trial actually failed to reach the clinical endpoint. So no patient benefited. And that's why they stopped the trial and completely abandoned this whole program, completely shut down their vector production facility in North Carolina.
So,
Okay. They spent $500 million to create this, facility. I mean, it's just what a sad.
so if I can make one comment, because
Sure.
I did work on DMD initially with Uta Franke in very early days.
Oh, okay.
So I know a little bit about DMD and how serious this is and
Yes.
so
Yeah.
the problem in many of those, not only, so I'm going to say both the positive and negative parts here. The
Yes.
positive part is that that we have come a long way since the initial cloning of, this is one of the largest genes.
Yes.
And making a vector, making it much smaller, kind of
Yeah.
mini gene that can complement. The trouble is that, as you said at one point, this is mostly, you can do that based on model systems,
Yeah.
and humans are not larger mice, and
Right.
there's not only the immune system, but something that I have been saying for eye diseases, but also in general terms, wherever whoever wants to listen, is that we have anywhere from 3 to 10 million variants in our genome, and we used to think they're all junk DNA or dark genome or whatever, and
Yeah.
that's really highly important for making us so unique that we are. And the implications of those variants we don't understand on disease, how they influence the disease actually trajectory is not clear. So whenever we are doing any treatment, we just base it on what is done in mouse or
I know. Yeah.
and nothing seems to have worked. And many of these companies, and now I'm actually one of those few people who is very generally very anti-big pharma. So I'm going to
Yes.
tell you my take on that, that some CEO or some scientist there goes after something without really doing deeper basic research.
Yeah.
So I want to hear, because you are one of the rare people in AAV field who has done the basic research on AAV. And most people really just sort of use it as a vector and
Yeah.
evolve as a vector. It didn't come up as a vector. So
Yeah.
I think there are different kind of genomes that evolved as vectors. Actually, there are transposable elements which are
Yeah.
like vectors. So it's just sad that they just go after this without doing any basic research. That's why most of these drugs fail. And
Yeah.
that is why the price of these drugs is so high, because they say, oh, we had to try 150 things to get one. You guys are stupid that you don't spend 1/10 of that money in understanding how the disease is and what's going on before going after this. This just a bhed. That in India we have the sheep thing, that bhed chal or whatever, every sheep is going to go out. So that's what is happening in the field. And that's why the cost of these is
Yeah.
3 million, 4 million, 10 million, because we didn't spend 1/10 of that in having the foundation. So that's
Yeah.
the problem. So I'm not against some of these things. You were saying, oh, Anand may not agree. No, I fully agree with the basic premise of this. What I
Yeah.
disagree is the way pharma deals with this and goes after. Same thing happening in retina field with complement drugs that
Yeah.
FDA is approving. That doesn't mean those things work many times.
Yeah.
So anyway.
I could not agree with you more, Anand. Yes. So hopefully, at the end, you'll get, where I'm going with this. Okay.
Can
So,
I
yeah,
can
go
I
ahead. Yeah, sure.
Yeah, I mean this is good because this is one case where somebody gets excited about patient vomiting compared to the previous patients who died. That really raised a couple of questions for me. Okay, one question is unconnected to this. So, this the term cure, do you think it
Yeah.
is appropriate in all those cases? Because do we know that AAV stays on forever in these individuals? Isn't it lost in some cases, like it happened, I believe, in hemophilia A trial? I don't know if that was one of the 14 that you listed
Yes.
or not.
Right. Correct. So,
And
yeah.
the other thing I've learned by sitting in, so Kalpana and I run a virology course along with Karthik Chandran. We have you may know him, Jayanta Rai Chowdhury.
Oh, yeah, of course. Yeah. Yeah.
Yeah, he teaches, use of viral vectors.
Yes. Yes.
So, and he has been talking about for a while now using AAV vectors for CRISPR-Cas9, and he believes that's the only context in which AAV can help in the sense of eliminating a disease, because after the CRISPR-Cas9, the virus is gone. And also in the class, he teaches about fragments of AAV integrated in the genome and causing
Yeah.
cancer. So
Great.
none of these case issues were a problem for those 14 diseases that you said were, taken care of.
So, again, depends, if you look at Luxturna, which is for Leber's congenital amaurosis, these patients, they've regained their vision, and the trial was actually, even the study was published in 2008, obviously trials were done earlier than that. So it's been coming up almost 10 years. Same thing with hemophilia B, more than 10 years, they're still expressing therapeutic levels of the protein. Yeah, hemophilia A, the vector was, and hopefully that will actually answer the question that you raised. Yes, over a 5-year period, the factor 8 level expression dropped from 100% down to less than 5%. And hopefully, so that's pretty prescient on your part. You're already pointing out the problem we have, and hopefully this will answer that question. Okay, so back to Sarepta and AVR74. So they were very excited, they decided that they were going to file for market approval, and that's where they came up with this AVR74 drug called Elevidys, and this was approved last year. Okay, the problem is this drug actually failed a phase 3 trial. So this is the first example of an FDA-approved drug that does not work.
It doesn't make
So-
sense. If it failed in the phase 3, how did the
Yeah.
FDA approve it?
No, because you see, other thing I should also mention, perhaps if you look at it from the point of view of the patients and their families, see, these are very rare diseases, and there is no other option. These patients have no other, way of, at least, so these people are, desperate to try anything. So they are pushing for it, and the FDA, felt
But
that.
if it doesn't have the endpoint, expected endpoint, what is the point?
No. So the FDA approval was granted, and the trial failed afterwards. So they had already gotten the approval because it helped some patients. A subset of patients actually benefited. But when they go to a larger trial in phase 3, it did
So
not
it's
work.
a statistical
Yeah,
thing. Okay.
correct.
Yeah, but there's another aspect of patient foundations. They play a major role in
Yeah, I agree. Yeah,
their
yeah,
case,
right.
whether they're effective or not, just because they want something to be there and somebody.
They, the good example is that brother, who was just desperate to try to help his brother, So these patients, I can tell you, I've talked to several families, they want to try anything because
Yeah.
doing nothing is not an option for them because, the shorter life
Yeah, just
expectancy.
imagine your brother is going to die
I
in
know,
front of your eyes.
I know, I agree, but they didn't know that then. Okay.
Yeah, Arun, I have another question, maybe dumb
Yes.
question. Are
No,
there no
wait a minute. There is no such thing as a dumb question. There's only a dumb answer. Yeah, go ahead.
Correct. Yeah, I also say that in class. Anyway, are there no human-derived serotypes?
See, you are about 30 minutes ahead of us.
Okay.
Okay, so I'll get to that point. Okay, so the question was, why is this happening? Why are we seeing, these kinds of problems, which we don't see in monkeys, right? Why we only see this in humans? Again, I will emphasize that the first generation of AV vectors are not optimal. They did not evolve to carry therapeutic genes. And of course, these are very, very high doses, and I'll go through these numbers at the end. So that's a problem. And you just said that, these are all monkey-derived viruses which were made into vectors. So they don't have tropism for humans. So we are just forcing this to, and hoping against hope that this will work. And this is clearly not going to work. And just like Anand said, these companies don't care about basic science. They are in it for making money. Okay, these are all multi-million-dollar drugs. They're all rich. They don't care. They do not understand basic science. They don't value basic science. So this is a problem. These are not based on scientific decisions. These are based on business decisions. Okay. So anyway, sorry, go ahead.
I may not agree with your statement that these companies don't care for basic science. They do care for basic science, but up to a certain extent.
Okay, I stand corrected. You're right.
I think we can have a long discussion on that someday. I actually completely disagree with that.
Really?
I know so many examples where they have ignored basic research and made the decisions. And I'm talking about big pharma. I'm not talking about the biotech companies.
Yeah, right. Okay.
Biotechs are great because there the real scientists are involved in doing that. But once it goes there, their purpose is venture capitalists want to
They
make.
they just want money. They are only in it
They
for the
don't
money.
really care about
Yeah.
So
Okay.
I think that I was talking more of big pharma business decision run by
Yeah.
the lawyers and MBAs
Yeah.
who have very little interest in doing this.
But also, Anand, the humans are so heterogeneous. I think if you want to do the basic research, you have to do for individual patient to understand completely, and that is never going to be a end of that kind of thing.
That's
So
excellent.
somewhere you have to call it. You have to take it and see. Okay, you have this much. Is this amount of information enough to treat a disease, make somebody's life meaningful? That is the question.
Yeah.
There is no end of basic science. Basic science keep on going until all the T's are crossed and I's are dotted. But that will, by the time, many diseases will, many patients will suffer. So that is the thing.
Yeah. See,
So
that's another very important point you raised here. So we start out with all inbred mice, inbred strains of mice, which are genetically identical. No two humans are alike, right? So that personalized medicine come into play, and all that stuff we can discuss later. All right. So it so happened about 3 years ago, 2021, the Sarepta team was visiting the University of Florida. They were very high on RS-74. This is very early stages. But they also, I think, knew something, was up. So they came to visit us, and I had an opportunity to meet with them. And they asked me a very simple question. They said, can you make AAV-RS-74 vectors better or more efficient? And hence the name of the title of my talk. And my immediate response was, yes, we can. Okay, and of course, as you know, Mr. Obama stole that line from us. Now, I'm just kidding. That's a joke. Okay, so why did I say that? This optimism was actually based on the fact that we had taken AV2 vectors and made it better. Okay, now this is all published data, so I won't belabor that point. So I'm just going to very quickly show one slide. So this is what we are calling the next generation of AV vectors. And actually, there are three categories. So when we modify the capsid, we call it the next gen. When we modify the genome, I'll go over that, we call it generation X. And we, of course, when we combine the two, we're calling it optimized vectors. Okay, so this is what we had done with AAV2. So I was fairly confident we can use the same strategy to make AAV-RS-74 vectors more efficient. So all you need to do, again, I won't go into the details because of the time limitation. All you need to make a couple of changes. In this case, 3 amino acids on the capsid were changed from tyrosine to phenylalanine. So we call this next generation vectors. And this we published in 2008. So these vectors are approximately 10-fold more efficient than the naturally occurring capsid vectors. So this means that if you increase the efficiency 10-fold, you can lower the dose 10-fold. That was the whole idea. And again, I'll go into the details of this one a little bit. We modified the genome. We call it Generation X. And this we published in 2015. And now these vectors are approximately 8-fold more efficient in expressing the transgene. And of course, you now combine the two, which we are calling the optimized vectors. So both capsid modification and genome modification. So these optimized vectors, you basically get an additive effect. So it will be approximately 20-fold. So this we published in 2016. And this vector is 20-fold more efficient. So we thought, well, this obviously is a rational approach to lowering the dose. So we use the exact same strategy for AAV-RS-74. And we published this last year. And the problem was the increase transaction efficiency was only about 6-fold, which is still better than, I always say, any fold increase is better than no fold increase, right? So
this is in the cells or in the animals?
In both. So we did in both human skeletal muscle cells and mouse muscle cells in vivo. So we can basically lower the dose. Even 6-fold reduction in dose would be beneficial. Okay. So how did we do this? So we established that, yes, we can do this, but that's not enough. 6-fold is not enough. And so what else can we do? So the question we asked was, can the extent of transgene expression from optimized RS74 vectors be further increased? So remember, RS74 vectors will go to the muscle. Almost every muscle in the body can be transduced. But the problem is that's only half the battle. It's the genome that does not express the transgene, especially in cardiac muscles. There's very little expression. So this is the crux of the problem. And this is something that has been completely ignored by everyone. So if you remember, go back to the AAV genome. AAV genome is single-stranded, right? We all know this. And again, these are the structural features of the ITRs. And AAV actually evolved to not express its own gene because it works great for AAV. AAV does not want to create problem for the cell. It just sits there quietly without expressing its genes. So when we add our therapeutic gene, we begin to expect to express our therapeutic protein to high levels. That will not happen. You know why? Because there is no host cell RNA polymerase that can transcribe a single-stranded DNA. So all RNA polymerases, transcription factors require double-stranded DNA. So unless it undergoes what is called the second-strand DNA synthesis to become double-stranded, this is completely a dead vector. That's why you need higher and higher doses, because you're trying to compensate for very, very inefficient. So I've always said this to my students, you cannot defy biology. If there is no RNA polymerase and you have a single-strand DNA, nothing is going to happen. So we decided we need to do something to boost expression from a single-stranded DNA. So how do we do this? So again, when we make a recombinant vector, I mean, it's kind of amazing to me that we are in some cases we are getting therapeutic levels even from single-stranded vectors. So the main culprit, the AAV, evolved over millions of years of evolution. It decided to create to remain single-stranded because it has. So if you go back to the ITR structure, so we have this 145 nucleotides, which is 125 of which are present as the T-shaped hairpin, and the remaining single. Why does AV carry this 20 nucleotide sequence? So if you go by this, so this is A prime complementary sequence. We have B prime loop, then we have C prime loop, and the remaining 20 nucleotides, which is present as single-stranded, is called the D sequence. Okay, and AV carries this D sequence for a reason, right? And so we figured this out, and I again won't bore you with all this is all published data. So we decided to focus on the D sequence. And it turns out that these 20 nucleotide sequence plays a remarkable role in the life cycle of both the AAV biology and also in the vector. And I'll try to give you a couple of examples of this. Okay. So a brilliant postdoctoral fellow joined the lab back in 1995. He was with me for 5 years, and he published this paper in PNAS in 1997. And he showed that the reason AAV vectors are very inefficient, because first of all, they're single-stranded. So why doesn't DNA polymerase take over and begin to synthesize the second strand? It turns out that the AAV has evolved a very interesting strategy to not allow second strand synthesis to occur. So Dr. Ching actually ended up identifying a cellular protein, which he named single-stranded D sequence binding protein. And this protein binds very tightly to the single-stranded D sequence at the 3' end and simply blocks the second strand. So DNA polymerase cannot get in there and extend this. Okay, so that explained why single-strand vectors are not very efficient. So, and then another brilliant grad student joined the lab in 2008, Chen Ling. He said, why don't we just get rid of this D sequence? So he replaced this D. So these are the 20 nucleotide D sequences at both ends. So he actually removed one of the D20 sequences and replaced it with a computer-generated substitute sequence, which he called the S20 sequence. So this is same length, obviously, but it also the same GC content. So try to make it very similar to D sequence, except no homology to the D sequence. And he asked the question, would this vector be better? So he actually called this generation X vectors. And, and I showed some of the data earlier on this. And he showed that, yes, if you use this vector now, you get actually 8-fold increased transgene expression from single-strand vectors, both in tissue culture cells in vitro and following tail vein injection in mouse hepatocytes in vivo. So again, the point is that using generation X vectors, you can reduce the vector dose 8-fold. So we decided to stay focused on the D sequence because it is doing something we didn't understand what was going on. Turns out that his wife, Yuan Lu, who also came with him, but she spent 1 year in the lab because she didn't get into the PhD program until 2009. She made a remarkable observation. She simply looked at this D sequence, which is shown here. So this is 145 nucleotides ITR sequence, and the last 20 nucleotides is shown here in red is the D sequence. And it's amazing, and I never would have guessed this. So she simply eyeballed the sequence, and she said, there's something interesting here. The sequence looked very familiar to her, and she noticed that this GTT-CCT, the last 6 nucleotide sequence in the D sequence, actually was very similar, if not identical, to the consensus glucocorticoid receptor binding element site, or GRE site. So the half GRE site is TGTTCT, which is GTTCCT. So I said, that's not very convincing. It's just happenstance that you see this. So this is a long story. She went on to show that no, this was a real functional GRE half site. So her husband Chen Ling said, well, went on to focus to show that yes, actually this responds to glucocorticoids. So and activates the glucocorticoid receptor pathway. Then the GR, which is present in phosphorylated form in the cytoplasm, enters the nucleus, binds to the D sequence, which is essentially the half GRE site, and promotes transcription, even from a single-stranded vector. So, and he presented this at the ASGCT meeting, American Society for Gene and Cell Therapy, in 2016. This is a long, so it took a long time to complete this. So he showed that yes, AAV infection actually activates the GR pathway, and the GR goes in and then binds to the D sequence and increases transcription. So then his idea was, if half GRE is doing this, why not replace the half GRE with the full-length GRE? Okay, so he thought, well, if you put the full-length GRE, now we get even higher level of transgene expression. So this is what Dr. Cheng did. So it's an interesting story. So he came as a postdoc in 1995 and was with me for 5 years, and then decided to go join Eli Lilly and Company. So he brought AAV to Eli Lilly, and Eli Lilly is now fully on board, spending billions of dollars and trying to fund companies and acquiring companies doing AAV gene therapy. So so he worked there for 18 years and then took an early retirement and said, can I come back and rejoin you? So I was only very happy to have him. So he's my lab manager now. He came back in 2018. So what he did was he simply added the GRE element. So I mentioned that the D sequence is actually, first 10 nucleotides are crucial for packaging. So you cannot really get rid of both D sequences. You can only get rid of one D sequence. But anyway, he put the GRE element downstream from the D10 sequence and put the transgene expression cassette. So he called it generation Y vectors. So we had generation X, now we have generation Y that contains the GRE element. And he showed that, yes, if you simply add this 15 nucleotide sequence, you get approximately 2-fold increase. Remember, this is already active because it is a half GRE site. And adding full-length GRE does improve significantly, although not quite as much. And they both respond to dexamethasone, which is known to activate the GR pathway. So, and this is AAV2 in tissue culture cells. So then he asked the question, what about if we inject this into mice? So again, wild-type AAV vectors don't transduce mouse hepatocytes very well. And now you see, just adding that 15 nucleotide GRE element Now we have 6-fold increase in transgene expression in the mouse hepatocytes. And this is not because we are adding more, injecting more vector DNA, because if you look at the total DNA copy numbers, they are not significantly different. So under this identical condition, same number of vector genomes, you're getting higher level of transgene expression because the GR is promoting transcription. So, so this paper was published, took a long time, but this paper was just published recently. And again, we showed that generation Y vectors can increase transgene expression 6-fold, and this is all AV2. So they can, therefore, we can reduce the vector dose by 6-fold. So another grad student, Jacob Sharty, who actually just graduated, this guy is amazing. He got his PhD in only 2 years. So he said, well, why are you wasting your time with AV2? AV2 is not going to be useful. So let's put this into AVR74 vectors, right? So he put GRE element in the AV2 genome, but packaged into AVRS74 capsid. And this is what he found. So this is transduction of primary human skeletal muscle cells in vitro. So this is what is happening. This is what Sarepta is using to achieve expression of dystrophin gene. So of course this is not dystrophin gene. This is GFP. So you can see this is the very, very low level of transduction, and then generation X does improve approximately 4-fold, and generation Y increases 6-fold. So just imagine if this was dystrophin gene, patient would actually benefit by this level of gene expression. And again, the vector genome copy numbers were not different. So this gene increased transgene expression is occurring because of the GRE element. So this is mouse, this is human cells in vitro. What about mouse? And again, Dr. Ching did the injections. You can see this is the gastrocnemius muscle cell, which is in the leg. And this is the tibialis anterior muscle. This is wild type. This is following intravenous injection of AVR74 vectors. And 8 weeks later, this is the level of transgene expression. And under identical conditions, if you now use the generation Y vectors, you can see approximately 20 to 30-fold increase. So this would definitely help the patients if this was a dystrophin gene. So this paper was...
Arun,
yeah.
how long this expression is maintained?
Oh, so we carried these mice for 6 months. I mean, obviously there was, so expression level stays. Yeah, it doesn't go down.
Okay.
So this is going to be very useful. So again, this paper was published last year, and we now can get up to 30-fold increase transduction, and therefore we can reduce the vector dose 30-fold. So, so this work was actually supported by Sarepta, and they were very excited about this. And said, well, we said, no, we can do even better. So Jacob Sharty actually, because he was interested in muscle gene transfer, he said, can we now use, he actually made a second set of vectors. So, we had generation X, we had generation Y, we have generation Z, and now we have run out of alphabets. What do we do now? So, so he actually not only added GRE, which is the generation Y vector, he also, again, I won't have time to go into the details of it, he also modified the A stem of the hairpin structure, okay? And he also added a muscle-specific promoter, and he called, because he made deletions in the A, so he calls it deletion A or delta A vectors. And then he packaged this into AVRS74 vectors. And he presented these data last year at the ASGCT meeting, and he showed by simply making this modification, now you can get 30-fold increased transgene expression. Remember the previous slide I showed you, that was with packaging, so just the genome and the packaging into the next-gen capsid. This is just simple modification here gets you 30-fold in the wild-type capsid. So again, you can reduce the vector dose 30-fold. And Sarepta was very excited about this. And so then when he tested this delta A vectors, again, this is primary skeletal muscle cells. You can see this is the level of expression, not very impressive. But just by modifying the ITR, you can see about 34-fold increased transgene expression. Again, if I were the patient, I would want this level of dystrophin expression in my cells. What about when we inject mouse muscle? And this really blew me away. Look at this. This is the level of expression we are getting with the wild type AVRS74, and this is the delta vectors, which is 30-fold increased transduction. So, and this is, remember, this is Elevidys, which is FDA approved drug. You know, I told Sarepta knew this data because they supported this research. I said, don't go, and they were getting ready to go for FDA approval. And I said, no, wait, and, you know, let's work on this and we can make this better. And, you know, there's too much money involved, and they went ahead and this vector failed. So anyway, they are now currently evaluating this in-house with this, and I think they will go for this. So this delta could actually become an FDA-approved drug because clearly this is so far superior to the currently used wild-type AAV capsid. And remember, this is just the wild-type capsid. Now, if we package this into next-gen capsid, we can at least expect to see a, you know, additive effect. So
Why didn't you do that one?
Sorry?
Why didn't you put in the RH vector and then see what kind of effect?
Yeah, we're doing that right now. Remember, this is all very recent, you know, just last year. So and Sarepta is you know, doing this with their dystrophin gene. So I'm really optimistic that this would work, and so they have licensed it from us, and hopefully, I'm really hoping that this could become an FDA approved drug. So therefore, patients would benefit.
Arun, can you comment on the level of expression of these proteins? Because it's one thing to have a certain level of expression, but you need
Yes.
optimum level of expression to really get the functionality you're seeing.
Yeah, I completely agree, Kishore. So, you know, this is one of my major limitations. You know, I'm just a vector guy. You know, I don't have a therapeutic gene. I don't have a disease model. So all we can do is reporter genes. No, you're absolutely right. This needs to be, you know, evaluated and corroborated by others using therapeutic genes and disease models. That's what Sarepta is doing right now.
So I think one more issue, I think,
Yeah.
that has to be considered in all of these things is that
Yeah.
this is active all your life, and unless you have a right kind of promoter and
Oh, yeah.
level
Yeah.
of expression,
Right.
and expression of RNA and expression of proteins are also two very, very different things.
Yes, correct.
You know, you have question of stability of RNA, stability of protein, and all that stuff.
Right,
So
right.
you see, that's where I say the basic research. We have
Yes.
sometimes no idea, and
Yeah.
we just do it. And overexpression of a protein can kill the cell after a while.
Yes, correct.
Overexpression of RNA doesn't mean overexpression of protein.
Yes.
And less expression of RNA doesn't mean less expression of protein. So I
Yeah.
think that's where the problem comes up.
Yeah.
That's a simple
You're-
research.
yeah.
If you do, you can save millions and tens of millions of dollars in the future.
Yeah. No, you're right. So, you know, Sarepta was insistent that we also look at RNA. I say, you know, there's only 40% correlation between our levels of RNA expression and protein expression. But they said, no, you have to do this. So they forced us to actually do this. And we did delta analysis. Yeah, RNA goes up, but, you know, it means nothing unless protein is expressed. So, and clearly they're not getting enough protein expressed. And, you know, I should mention this, that this promoter that we use is called a CK8 promoter, which is highly human muscle specific promoter. So we believe that if this was true, if this was dystrophin gene, the patients will be expressing higher levels.
Hey, Arun,
protein. Yes.
In that GRE mediated transcription, what is the
Yes.
RNA polymerase involved?
No, GR is completely promoter agnostic. It doesn't matter, you know, any transcription factor or any promoter, any polymerase would actually be activated by GRE element.
Mm-hmm.
It's a global, you know, enhancer of transcription. GRE element is.
No. I was referring to the fact that it was a single-stranded template.
Yeah, so actually single-stranded works because, you know, it's not optimal because in double-stranded actually it's much better. So we also have double-stranded vectors that GRE element get dramatic improvement transgene expression.
Okay.
So anyway, so as I
Arun,
mentioned,
I have another question.
yeah, go ahead.
the side effect you saw in the first generation vectors,
Yes.
is it due to the dose of the vector or it is something else?
Yeah. Can you
You
give me
have a
5?
correlation with the
Yeah.
increasing dose, you will have increasing
Yeah.
side effects.
Okay. Give me 5 more minutes and I'll answer that question. Okay. All right. So here I'm not going to bother you with all the summary, but just the last point. I believe that the optimized opt-wide, which we call opt-Y and opt-delta AVR74 microdystrophin vectors are likely to lead to therapeutic correction of the Duchenne muscular dystrophy at further reduced doses, which is, I totally believe in basic science will answer these questions. So, I give this talk and so far no one has disagreed with me. So especially to my clinical colleagues, I say I have a few humble suggestions for you. I'm a basic scientist. I cannot do clinical trials, right? And also make this little joke, I don't know how this happened. I forgot to go to medical school. It just completely slipped my mind. So, and again, as I said, no one has so far disagreed. So I say we should not be using the first generation of AV vectors. Please stop using this because AV did not evolve as a vector. And I say you should be using capsid plus ITR modified vectors in all future clinical trials, right? And definitely use lower doses because I think, and as you will see, it's a vector dose that is causing the problem. And of course, you don't want to compromise on therapeutic efficacy. But the most important point I try to make, and as some of you already guessed it, we really should be using human tropic AV serotype vectors. So the two companies that I actually started were based on the fact that these vectors do not transduce mouse cells. So that's why they were ignored. And now we have two vectors that only transduce primary human cell types. One is liver and one is hematopoietic stem cells. So that's why I think there's much more likelihood of success if we use a human tropic serotype vectors. So again, after the first 4 patients died back in 2020, there was a serious question raised. AV vectors, are they safe? And I really felt compelled to write a letter to the editor of Human Gene Therapy, which was published in July issue of this journal, where I actually wrote, it is quite a testament to the remarkable safety of even the first generation of AV vectors that humans are easily able to tolerate roughly the same number of AV particles as their number of cells in the body. So actually it turns out, and I'll show you those numbers, you can actually inject up to 100 trillion particles in humans, and you will see no adverse events, okay? But if you cross that threshold, if you go over that, then you run into problems. So my suggestion was that use capsid ITR modified vectors that are human tropic. They are less likely to be immunogenic and most effective at lower doses, thus further increasing the safety and reducing vector production costs. So here's the problem. Vector production cost is astronomically high. That's why the price is so high. And then because they have to make large amounts of it. And I think Prasad mentioned this, the hemophilia A vector, which is AV5 for him, that BioMarin was making. They inject such high dose because they have to produce large dose. So the and it didn't work. So they have actually they have called it a commercial flop. They have completely stopped the trial and they're no longer producing the vector because the cost is so enormously high. And there's again, if you use human tropic vector, that will almost guarantee to ensure translation to the clinic with higher probability of success. So
that means they had dual problem. On the
yes,
one hand, they use very high levels of virus. On the other hand, at the opposite effect, namely, they could not detect the AAV after a while.
yes, right. So that's very insightful point you made. So there are two problems actually. So BioMarin was using AV5, which actually transduces human hepatocytes very poorly. Okay, so that's one problem. So this is again going back to the basic science versus business side of things. they only could get exclusive license to AV5 because everything else was taken. So they went with AV5. I mean, that's a wrong way to make a decision. And then the problem is, so factor 9 trials are very successful, even with the AV5, because factor 9 is made by hepatocytes in the liver. Factor 8 is not made in the liver. Factor 8 is made in liver sinusoidal endothelial cells. So they're forcing hepatocytes to make factor 8, which is a much larger protein, and hepatocytes have a very hard time secreting that because they're not designed to secrete large proteins. And the other problem is only 5% of the or so of hepatocytes are transduced with AV5. So very small percent of the hepatocytes are expressing high levels initially of factor 8. And then there's so much stress on those hepatocytes, they basically give out. And that's why you see a steady decline over 5 years, because the hepatocytes are gone. So we are actually we actually have a vector that will transduce endothelial cells. I think that is the answer. So using the wrong serotype and wrong cell target, I think there are two mistakes they made. But they won't acknowledge it. That's the problem with it. I actually gave a seminar and they listened to me, and then they completely ignored it. So anyway, what can you do?
Arun?
All right. Yes.
So you may have mentioned this. Where does this human tropism reside in? And why was it so hard if they can do it in monkey cells, why couldn't they transfect human cells and try if those cells are receptive to this virus?
Yeah, so if they work, just because it works in monkeys, there's no guarantee it will work in humans, right? That's why I think our starting point should be to first test in primary human cell types, and then go back.
Why wouldn't regulatory agencies insist? Why don't you try in human cells? That's easy,
right? This to me that is amazing. I'm just a stupid guy, suggesting these ideas, and these are the brilliant people who are doing this work, and they don't seem to see. So, I had a very good friend. He was actually he's a hematologist, originally from Kerala, but he's more like a philosopher. And he, once told me, he said, the problem is what the mind doesn't know, the eye doesn't see. So, I thought that was a very profound statement he made. So I think, I hate to even say this, that I think we are doing it all wrong. And that's why we have run into problems. So simple logic would suggest that, don't expect, don't hope that it'll work, because hope is not a strategy. You have to think, coherently and think logically to design your vectors. Okay. So I'm just want to leave you with, what about the high doses? This has come up many, many times. So when I first saw the number of doses high doses that were giving, I'm not very good with numbers. So I wanted to know what those 1 time 10 to the 13 or 1 time 10 to the 14, what does that actually even mean? So this is a company called Unicure. They have an FDA-approved drug for hemophilia B, which is AV5. And they are injecting 2 times 10 to the 13 vector genomes per kilogram. So this is, on an average about 70 kilogram patient they are using. So I try to calculate. So if you do this simple math, 2 times 10 to the 13 vector genomes per kilogram in 70 kilogram person would translate to 1.4 quadrillion particles per patient. Okay. So I was very curious, how many cells in the human body? And it turns out that there is a paper published in 2016 by Sender and colleagues. They estimated the number of cells in a 70 kilo. It's amazing. This is the same 70 kilogram person, a reference human they called, is only 30 trillion. So what I did was I divided 1.4 quadrillion by 30 trillion. Okay, just to ask the question, how many cells, how many vector particles per cell for every cell in the human body? And that
It's
number.
a true virologist question. What is the MOI?
MOI, exactly. In vivo MOI, multiplicity of infection. So each cell is, well, of course, not each cell is getting it, but on an average, we are injecting 47 particles for each cell in the body. I think this is insane. There's way too many AV particles, and it gets worse. So now BioMarin, we talked about, they are using 6 times to the 13 vector genomes. That is 4.2 quadrillion particles per patient. Again, you do a simple math. That turns out to be 140 particles AV particles for every cell in the body. It gets even worse. Pfizer, where the 2 patients died, they were injecting 2 × 10⁻14 vector genomes per kilogram. That is 14 quadrillion particles per patient. That turns out to be 467 particles per every cell in the body.
That is just the total number of cells. If you only take the target cell.
Oh, that is much higher. Absolutely, yeah. It is just in my mind. It's mind-boggling.
It's a small
It is
fraction of
But
cells.
Arun, to be
Yes.
fair,
Yes.
unless you do the pharmacokinetics, this
Yeah,
is a mathematical,
I know. This is
Particularly.
yes, I agree. These are playing with numbers, but I'm just trying to understand the extent of the dose that is delivered.
Arun, simple question. What happens
Yes.
if you inject the same dose into a healthy patient, healthy volunteer?
Yeah, so that has been actually done, but not as high a dose. No, correct. You're right.
But
So
also
again,
depends how are
100.
you
If you
injecting.
inject if you inject 100 trillion particles, nothing will happen. That has been done in
But
phase
no,
1
the
trials.
question is, where are you injecting, Arun? The question
So
is,
that depends. For hemophilia B, which is Unicure, this is IV, BioMarin IV, and this is also IV. Everything is IV.
So
Also,
Duchenne is IV also?
yeah, everything is systemic in administration, right?
Yeah,
Of
because
course, I works because it's very limited amount, and it's immune privileged organ. It works in brain, so that's why eye and brain are the best organs for gene therapy. Systemic administration is problematic because you don't know where the vector is going, because none of these serotypes is human tropic.
yeah, brain is understandable because of the blood-brain barrier.
Yes, right. So
You want to
AV9
bypass that. But
AV9
DMD, why are they not doing intramuscular injections?
Oh,
at critical sites?
that has been done, but they did this. It's just muscle mass is so huge that it's very difficult to transduce muscle by direct injection. And you have to give this one trial. They did 150 injections.
Now,
Just
my other question is, do you
Yeah.
find AAV in heart muscle?
I'm going to leave.
Yeah,
Thank you.
AVR74 can get to the heart, but the expression level is very high, very low.
Yeah, because the reason I ask that is skeletal muscle is very different from myocardial muscle.
Yes, correct. You're
Because
right.
heart is made up of individual cells,
Yes, you're right.
whereas skeletal muscle is a syncytium.
Yes,
So you can pretty much, that's why you can
Right.
inject a medicine anywhere in the muscle and
yes,
it will spread
right.
throughout the skeletal muscle.
Yes, you're right. So
So
the
the reason I say that is
Yeah.
in the Pfizer study, the 2 people who died
Yes,
had cardiovascular
Yes.
complications, pericarditis, and
right.
so I'm wondering, even when you treat the patient through intravascular route,
Yeah.
it may not be reaching the cardiac tissue.
Oh,
So
it
the
is.
cardiac complications
Yeah.
of DMD,
Yes.
that's why I asked, what is killing these patients?
Yes. So
And
you are absolutely right.
pathology-
So yeah, cardiopulmonary insufficiency is a huge issue because the vector does get to they're using a wrong promoter, Sarepta is MSCK7 promoter, which is skeletal cell specific, but not
Yeah, typically
cardiac
you
tissue.
see Coxsackie viruses
Yes.
affecting the heart,
Yeah,
Coxsackie
correct.
A and
Right.
B.
Yeah.
That's why that was going to be my next question. Is there any possibility of using those as vectors?
So that I don't know, but I can tell you the CK8 promoter that we are using expresses extremely well in the cardiac tissue. So I think that's the other thing that's what we need to do. Okay. All right. So the first 4 patients that died in the company called Audentis for myotubular myopathy, they were using 3 × 10⁻14 vector genomes, and that is 21 quadrillion. I think this is the uppermost limit that has been used, and this is so if you calculate, it turns out there are 700 times each cell was getting the AV vectors. So, and this is where the problem began. First 4 patient death occurred in this Audentis trial. And then this is Zolgensma, which is the FDA-approved drug for spinal muscular atrophy. This is their standard dose, 1.1 × 10⁻14 vector genomes per kilogram. This is Novartis bought this for $8.7 billion. They injected this into a 4-month-old baby. And I don't know how many cells a 4-month-old baby is, so I looked it up. So you can calculate based on the weight of the 4-month-old baby. I assuming it's, rough approximation. So I did the calculation. So this 4-month-old baby was administered with 770 trillion AV particles. I mean, this is just insane. I mean, any, rationally thinking individual will see that you cannot do this. And this was the first death that occurred with an FDA-approved product by Novartis. And
This
two
is for
more.
which indication?
This is for spinal muscular atrophy. So these children are born with completely paralyzed because there's a protein called SM1 protein that is missing in the brain. So AV9 was used by IV injection, and AV9 is the only serotype that can cross the blood-brain barrier. And yes, it can, but at a very, very high dose. And the problem is this AV9 also transduces the liver very well. So liver is expressing this protein, and God only knows, 90% of the vector actually goes to the liver, only about 3 to 10%. I think that's an overestimate. I think it's more like 1 to 3% of AV9 gets to the brain. The good news is that, yes, there are about 30 patients. So average life expectancy of SMN patients is about 1 to 2 years. And these are now healthy 4 or 5-year-old. They're running around. So, and there's no other treatment available for them. So yeah, the patients are willing, families are willing to try this. But clearly, now there, as far as I know, 3 patients have now died with Zolgensma, which is this drug. Okay.
Isn't Arun, isn't there
Yes.
antisense oligo for
Yes.
muscular
Yes.
dystrophies?
Yes. So it's called I forget the name. Yeah. So but the problem is you have to inject every week or every 2 weeks. And that, so the quality of life is not, I mean, if you can cure the disease by 1 injection, that would be preferable, right? So yeah, I know what you're talking about, but I don't remember the name.
Yeah, that's
yeah, you're right. Yes. Okay. So I'm just going to leave you with one last slide. A final word about the AV serotypes that are human tropic. Okay. So a few years ago, there's a guy named Phil Johnson. He used to be at Children's Hospital of Philadelphia. He came, I think he's currently a CEO of a company that he started called Interius Biotherapeutic. At one of the American Society for Gene and Cell Therapy meeting, he said when it comes to gene therapy, we use mice and then maybe monkeys and then go to again, I mentioned this many times. So he actually showed a slide. He said, when it comes to gene therapy, mice lie, right? And he said, monkeys tell white lies, and only humans tell the truth. So unless so he actually suggested that let's forget about all, animal experiment, go straight to humans, do the clinical trial, especially in diseases where the life expectancy is known to be short. And at least we'll get the right answer, because no matter how much time, money, and effort you spend on working with animal models, there is no guarantee that it'll work in humans. So that idea was soundly rejected by the FDA. So he switched to now he's working with lentiviral vectors. So when I saw this, I really like this actually. So I said, well, I should update this. I've actually done this. I have colorized this. So I said, when it comes to gene therapy, mice lie, right? Monkeys tell white lies, and humans tell the truth. You know who this guy is, right? Is alleged to have never told a lie. This is the first president of the United States, George Washington. So I said, yeah, okay, this makes sense. But I am not so sure now. You know why? Because of what has happened recently. So
Yes, this may be the right one.
right. So this man is even capable of telling the truth. What was amazing to me, that the remarkable similarity between the two, right? Look at this. So I don't mean to offend anyone who voted for Mr. Trump, but I think, we really need to know the answer of what happens in humans. So,
but also, Arun, if I may add,
yeah,
human also, not every human tries.
exactly. That is true.
So
You're right. Yeah.
I think there is so much of heterogeneity.
Yeah.
I think that's where the personal medicine comes into.
Right. So I don't know. So my next door neighbor is a strong, very strong supporter of Mr. Trump, and so we had a bet. I really thought that Kamala Harris will win because I mean, you don't want a convicted felon in the White House, right? Anyway, so he made me promise that if Mr. Trump wins, I must wear the MAGA hat, right? So I reluctantly agreed. So of course, when I agree to doing something, I do this. So I said, okay, I will wear a MAGA hat. So I made a MAGA hat actually. I don't know if you can see this. So it says, "Make AV great again." So it is still MAGA. So he agreed with me. All right.
Okay, Arun, we
Yeah.
had a group of 11 people here. We also had the bet. Myself and Mohan, they said
Yes.
Trump will win.
Oh,
All of us,
really?
Hillary will win. And
Mm.
we were right.
Yeah. Anyway, I just want to thank everybody who did the work. Dr. Ching, who went to Eli Lilly and Company, has now come back. Chen Ling is a professor at Fudan University. His wife is a scientist at Full Circle Therapeutics in Shanghai. Jacob Shardy finished his PhD and is doing a postdoc with me. Anisha Nath is a postdoctoral fellow, and Jeff Keeler, actually, he's the guy next door neighbor who helped us with this. He's a nice guy, but he's a MAGA guy. And these are some current lab members, and these are the collaborators. And these are the funding agencies that have funded us for over the years. And some of the work that I presented today was supported by Sarepta. So finally, I just want to dedicate this presentation to the memories of the father of AV vectors, Nick Muzichka. Sadly, he passed away last January 6, 2023. And sadly, the grandfather of AV, Dr. Ken Burns, my former mentor, passed away earlier this year on January 26, 2024. So after Ken, I mean, he brought AV to the University of Florida, and he was, true visionary. And when he passed away, the University of Florida has a magazine they publish called Florida Physician. So I thought they came up with the best name for him. They called him an amazing visionary, or AAV. So this is synonymous with AAV. So all I'll say is rest in peace, Ken. I'll stop here, and thank you very much.
Thank you
And
very
I
much
hope I didn't put you to sleep, but I'll be happy to answer any other question. Thank you. Sorry, were you saying something?
I was thanking you for coming back again and giving this wonderful. And the thing is, there is so much of discussion here.
Yes,
really makes
yes, right.
Okay. And thanks once again. And we are hoping to hear about your Navigen's progress too. So
Yes, actually, yeah,
yes.
I'm hoping that, I'm talking to a couple of investors. I know, I think Prasad mentioned CRISPR-Cas9, but, I actually am not sure CRISPR-Cas9 is the best.
I mean, CRISPR has its own utility in a different setting, and this is probably has a different utility, but I don't know whether you can combine those two and get better efficacy.
Yeah, if you have a couple of minutes, I'll tell you one short story. There's a guy named Matthew Porteous. He's at Stanford University. He was the first person to use CRISPR-Cas9 to correct sickle mutation. So, I mean, we've known about sickle cell disease since 1957, the mutation. And this was the first attempt that, that he was able to do this. And he published this paper in Nature in 2016 and generated so much interest that he formed a company called Graphite Bio and generated a lot of money. It went public, and he did the first trial, and the first patient that he dosed with CRISPR-Cas9 almost essentially died. So much adverse event. So last year he gave a presentation. He showed a slide showing that he said CRISPR-Cas9 is a fantastic tool. It works great in tissue culture cells. It works great in mice. It works great in monkeys. But the problem is that the human immune system doesn't know that CRISPR-Cas9 won the Nobel Prize in 2020. So human immune system, you cannot fool. Mice and monkeys, you can put anything you want and they will tolerate, but humans will not. So I'm hoping the nucleus-free genome editing that we are developing will probably be much safer.
Arun, I got to go, guys.
Yeah, I know. All right. Well,
You can continue. CK,
no.
if you
I
are
already taken too much of your time, but I greatly appreciate CS the opportunity that you have given me 4
you
times.
are most welcome. You are most welcome.
Thank you, Arun. It was wonderful.
Thank you, CK. It
Arun,
means a lot.
fantastic talk.
Coming
Yeah,
from you,
fantastic.
it means a lot. Thank you.
Oh, are you talking about me? I'm just a
Yeah.
layperson.
No. You have no idea how much I admire you, CK.
Oh, thank you.
No, seriously. Yeah. I mean, you're the